Influenza can dismantle the structures inside the nucleus of a human cell and use some of the released proteins to make more viruses. It does not rewrite your DNA. It does something cellular. It reorganizes the room where your genetic machinery works. A new study has now mapped how a virus with only a small set of proteins pulls that off. A study published in Nature Microbiology mapped direct contact points between influenza A proteins and human proteins inside infected cells. That inside matters. Many interaction studies remove proteins from cells before examining them. Here, the researchers use in-cell cross-linking mass spectrometry. They chemically locked nearby proteins together while the infection was happening, then identified the contact sites. The result was a molecular map of how influenza physically interacts with the machinery of a human cell. One of the most striking targets was a group of structures called paraspecles. Paraspecles are tiny membrane-free compartments inside the nucleus. They form around a long non-coding RNA called MEAT1 and help organize proteins involved in RNA processing, stress responses, and gene regulation. During infection, the researchers watched these structures progressively disassembled across several human cell lines. And this was not caused by one viral protein acting alone. Influenza nucleoprotein and NS1 contacted proteins normally concentrated in paraspecles. The viral protein packs degraded long non-coding RNA housed there. The infection also inhibited the cell's RNA polymerase II activity. Together, those changes destabilize the compartments. But here's the part that makes this more than simple cellular damage. When the paraspecles came apart, they released host proteins into the nucleus. Functional experiments indicated that some of those proteins helped influenza replicate. In other words, the virus may be dismantling a structure not merely to destroy it, but to gain access to useful parts. The study also identified host interactions involved in processing hemagglutinin, the surface protein influenza uses to attach to cells through the endoplasmic reticulum and Golgi system. So influenza is interfering with both nuclear organization and the cell's protein processing network. This does not mean the flu rewrites your DNA. It reorganizes cellular machinery. It is not editing your genome, and it does not mean researchers already have a new treatment. These experiments were performed in infected human cell lines. A host protein that helps the virus may also be essential for the patient, which makes it difficult to target safely. But a detailed interaction map can reveal dependencies the virus cannot easily replace, and those may eventually become therapeutic opportunities. A flu particle does not carry a factory of its own. It enters yours, changes the layout, opens the locked cabinets, and starts building with what it finds. For more science without the hype, subscribe to my channel of Science and Other Demons.